I work for the ICBF in the area of major genes and genetic testing, and I was asked this evening to give a presentation on genetic defect testing technologies and to mention a small bit about myostatin as well. So this might be a bit of overlap with the previous slides. So just a bit of background first. So typically in Ireland, a farmer will take a tissue sample, be it an ear tissue sample or hair sample from the calf at birth and send it off to the laboratory. In the laboratory, the DNA is extracted and there's a couple of sample preparation steps done on it before it's genotyped on a SNP chip. And I'll go into that in more detail, but essentially what happens is a genotype is provided for about 50 to 52,000 SNPs that we want to interrogate. And the service provider will report those results and upload it to the ICBF database. And from there, the ICBF can do a couple of things, like the guys mentioned previously. So we can confirm that the parentage of the animal, we can identify the breed composition of the animal. We can provide a genomic evaluation, beef or dairy. And then what I'm going to focus on is the major gene status. So what is a major gene? A major gene is any gene or gene variant that's known to be associated with a genetic condition or trait of interest. So that may be genes that are costly to producers, such as those associated with high mortality or embryonic loss, or genes that can be a good way to increase profits. So those that produce a positive effect on the animal's production. So maybe the pollenness or the myostatin gene as well. I suppose traditionally, a carrier animal for a genetic disease was only discovered by the farmer after it had produced an affected calf. And the farmer then typically had two options, either to cull that animal and its relatives, sire, dam, or risk producing another calf. So by genotyping, we can determine the major gene carrier status before any progeny are produced. And the farmer in turn then can make more informed breeding decisions, be it culling the carrier or mating with a non-carrier. So if we take scenario 1 here, if we say the capital A is the normal version of the gene and the small a is the affected version of the gene, and for instance, the farmer isn't aware of either major gene status of the dam or the sire, and he crosses these two animals, and he gets a 50% chance of producing a carrier animal, a 25% chance of producing an unaffected animal, and a 25% chance of getting an affected animal. If we take scenario 2 and the farmer is aware of the carrier status of the dam, they can take more care when picking a non-carrier bull. And so when you cross a carrier and an unaffected sire, then that'll avoid producing any affected offspring. So you'll have a 50% chance of getting an unaffected calf and a 50% chance of getting a carrier calf. And that's just one of the benefits of knowing that the major gene status of your stock and just getting the most genetic potential from your herd. Onto the genotyping technology then. So I suppose at the base of every technology we use in genomics is DNA, which is our genetic material passed on from one generation to the next. And what it is, it's just a long chain made up of nucleotides, which are essentially the building blocks of DNA. And the nucleotides can be one of 4, so they can be an adenine, a thymine, a cytosine, or a guanine. And we can abbreviate those A, T, C, and G. And A will only match with a T and vice versa, and a C will only match with a G and vice versa. And that's what we call complementary base pairing, and that's important in the next slide. And just for perspective, then there's about 3 million DNA of these DNA bases in the cattle genome. So sometimes when DNA is being synthesized, or copied, little mistakes can occur. So for example, where T is accidentally replaced with an A or a G or a C. And these are what we call single nucleotide polymorphisms, or SNPs, and we will refer to them a lot when we talk about genotyping. So for example, if you take the purple animal there and her string of nucleotides, she has an A where the blue animal has a G allele. So for example, if the purple animal, the A allele was a carrier for NT81 myostatin, the blue animal would be a non-carrier and wouldn't have the double muscled phenotype. So these SNPs are responsible for the differences that we see between animals. and they occur at a really high frequency in the genome. And for that reason, we can use them as markers for identifying genes associated with genetic conditions that we're interested in. The actual technology we use behind genotyping is called microarray technology. And basically that allows us to zoom in on areas of interest in the cattle genome. So if you take that plate there, each little square is a microarray housing the various different SNPs or genetic markers we want to interrogate. So for example, if we take our own chip, there's about 52,000 SNPs chemically bonded to each glass slide. And what happens is the DNA is extracted from the biological sample, be it the hair or the ear tissue sample. It's applied to a microarray. And using the same idea I mentioned in the previous slide of complementary base pairing, the animal's DNA binds to a complementary sequence in the sample DNA. A fluorescent signal is produced, and a machine reads the intensity of that signal as it passes through. And the intensity of that signal will indicate whether the animal has an adenine, a thymine, a cytosine, or a guanine in that position. And that's basically how a genotype is produced. So the ICBF have their own custom genotyping platform, or genotyping microarray, that we call the IDB SNP chip, or the International Dairy and Beef SNP chip. And that was developed back in 2013 initially, in collaboration with Teagasc and Weatherby's Ireland. And it basically gives control of the chip content we want to put on the chip that's important to Irish breeders, and how the chip is developed and who's involved in it. So there's been 5 different versions to date. V1 started out with 16,000 SNPs, and we're currently on V5 today, which is a Thermo Fisher, based on a Thermo Fisher technology, and there's 52,000 SNPs on that currently. So within that 52,000 SNPs, there'll be SNPs dedicated to imputation, to parentage, verification and discovery, a group of research SNPs. So we would give research groups they'd be able to submit research SNPs that might be of interest that we can screen for in an independent cattle population. And then finally, what's relevant here is the major genes. So there's about 165 major genes on the V5 platform, and they fall into categories of lethal, unwanted, colour, meat, milk, and beneficial. And I just pulled out a few that might be of interest to the Aberdeen-Angus Society, such as myostatin, polled, calpain, calpastat, which would be meat tenderness genes. And then we have the hydrocephalus, the fawn calf syndrome, curly calf, dwarfism, which would be all kind of lethal or lethal embryo major genes. And while they mightn't be that common in Ireland currently, we're always screening for them to see if they do segregate in Irish cattle. So again, the ICBF use this SNP chip internally for a multitude of purposes, be it parentage, providing genomic evaluations, and the coding of major genes. So arising from the IDB SNP chip is the major gene pipeline. So I suppose major gene genotypes are of huge interest in AI companies, breeders, and herd books. And the ICBF are currently in the process of implementing an automated pipeline which allows the large-scale reporting of these major gene genotypes. So we're currently working with V5 genotypes, so any animal that was genotyped with the V5 chip. And as I said previously, there's about 165 major genes on that chip. Essentially what it is, QC pipeline with the aim to increase reliability of the genotype, of the animal's genotype. So there's a number of QC steps that are involved. I won't go into the detail of them, but they're essentially measures of data quality and how the variants and how the genotypes are actually performing. And it also includes a Mendelian check for the sire, the dam, and the 3 animals where available. The end result is a confidence that the call is correct to allow automated reporting. So, if the pipeline is saying an animal is a homozygous carrier or a homozygous non-carrier, that the pipeline is reporting it as such. And that has multiple downstream benefits for the likes of yourselves, the breed societies, and we're now able to release the myostatin genotypes to go on pedigree certs for sire advice. So we want to be able to add, if a farmer looks up for a bull that he'll be able to see what major gene, if any, if that bull is carrying any, and to all genotyped animal profiles down the road as well. So this is just an example of a zootechnical certificate for a male Angus, part of the Irish Aberdeen-Angus Association. And if you look down there at 13.3, you can see that he's a myostatin non-carrier for 9 different myostatin variants. And that's how it's going to be reported, and is currently being reported. So there are over 60 major genes gone through the pipeline to date, and these 35 have been reviewed and validated, and if they haven't been released already, they're very near for release. As I said previously, the myostatins are already out there. The others include some Holstein haplotypes that would be important to the dairy, calpain, calpistatins, meat tenderness, some colour coat genes as well, and some unwanted down there at the bottom as well. And that list is constantly being extended. And I just pulled the frequency of these variants that have been through the pipeline in purebred Aberdeen-Angus animals. So you have the Pole Celtic there, which is at pretty much fixed there at 100%. You have the meat, 9 different myostatin variants there. So the 2 segregating are the NT81 at almost 3% and the F94L at 0.1%. Cold colour, then you have the black gene and the red factor. No surprise there. Meat tenderness, then you have the calpain and calpistatin. So they're segregating from 8.4% there up to 69%. And then at the bottom, we have the milk capicaseins, which may or may not be of interest to segregating there about 4% right up to 80% for the B variant. Finally, just to finish off on myostatin. So myostatin is the gene we associate with double muscling in cattle, and I suppose typically associated with improved carcass performance, better killing out percentages, higher feed conversion ratios, and increased average daily gains. And there's also improvements there with meat tenderness and the quality of the meat coming from these animals and the more desirable cuts of meat. On the other hand, then you have, increased associations of calving difficulties, dystocia, cesarean sections, which in turn have a knock-on effect on female fertility and, calf mortality and survival. So the gene itself is found on chromosome 2 in cattle, and as of this morning on the ensemble database, there was about 770 polymorphisms in the myostatin gene. So we really look at a very small amount, about 17 in total, and there's 8 to 9 that are actually segregating and having an effect. So in the Aberdeen-Angus, we said already, the 2 segregating are the NT81 and the F94L. And I pointed out the location there because it's quite important in terms of the effect the SNP has, and how the SNP goes on to affect the end protein structure. So, for example, if we look at Q204X, E291X, NT81, they're all quite damaging to the protein because of where they lie. And they would be typically more associated with double muscling, but also increased birth weights and in turn calving difficulties. On the other hand, then you have F94L, S105C, D1A2N. They're more conservative. They don't seem to damage the protein as much. They're associated with the increased muscularity without any change in birth weights and subsequently less effect on calving difficulties. So just to conclude then, genotyping can provide useful information on an animal's genetic disease status, allowing the farmer to make more informed breeding decisions. Microarray technology is the current technology we use to genotype, which allows us to zoom in on areas of interest in the cattle genome. IDB SNP chip is our custom genotyping platform within ICBF, and we use that for a multitude of services across multiple breeds. And major gene pipeline is currently being implemented to allow for large-scale reporting of major gene genotypes. And I'd just like to acknowledge the group of people that are involved. So I only joined ICBF back in February, so this group have been working tirelessly on that major gene pipeline and getting those genotypes released. So just want to acknowledge their input as well. So that's it. Thanks for listening.